I am Naqib Mehdi, and in this module we would be focusing mainly on inorganic plant nutrition, wherein we will be studying about the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. And you would also be learning about the role of the essential elements that are required by the plants. The origin of hydroponics can be traced back to the ancient city of Babylon, where present-day Iraq is located. The Hanging Gardens of Babylon was one of the 7 wonders of ancient world, is the first known example of soil-less growth of plants. This was around 600 BC. But the first standardized nutrient solution was created in the 18th century by Julius von Schaps and Wilhelm Knopp, 2 German botanists, which laid the foundations of hydroponics as we know it today. This solution made it possible to grow roots without any medium. Knopp went on to create yet another solution which is still in use today. The word hydroponic was first used in 1937, coined by Dr. William Frederick Grieke, aptly known as the father of hydroponics. He made news when he was able to grow tomato vines more than 7 meters long just using mineral nutrient solutions right in his backyard. The word is a combination of Greek words, hydro meaning water and ponos meaning labor. Let us see how a basic hydroponic setup works. Hydroponic plant production. Plants are grown in a tube or a trough placed on a slight incline. A pump circulates a nutrient solution from a reservoir to the elevated end of the tube. The solution flows down the tube and returns to the reservoir due to gravity. Inset shows a plant whose roots are continuously bathed in aerated nutrient solution. The arrows indicate the direction of the flow. For effective hydroponic setup, we need 4 things: a nutrient solution, substrates. In hydroponic crops, the plants draw the nutrients from the solution, but they still need support and the root have to be sufficiently aerated. Some of the most widely used substrate are perlite, rice husk, wood, fiber or wool. Hydroponic crop requires more technology and precision than a conventional one. Some of the necessary instruments and equipment are conductivity meters. The electrical conductivity of the nutrient solution indicates the amount of dissolved nutrients and whether they need to be replenished. pH meter. It is essential to control the acidity of the solution and substrate, as the optimal level is different for each crop. Lighting. Sunlight, artificial light, or a combination of both can be used to maximize yield. In recent years, the use of LED light has increased due to their low consumption. Air control. In closed environments, the concentration of carbon dioxide in the air can be increased to improve fertility. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. Few of the benefits of hydroponic includes the ability to produce higher yield than traditional soil-based agriculture, allowing food to be grown and consumed in areas of the world that cannot support crops in the soil, eliminating the need for massive pesticide use considering most pests live in the soil, effectively making our air, water, soil and food cleaner. One of the most important advantage besides the above is the study of plant nutrient research, and hence a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involves the culture of plants in a soil-free, defined mineral solution. These methods require purified water and a mineral nutrient salt after a series of experiments in which the roots of plants were immersed in nutrient solutions and wherein element was added, removed or given in varied concentration, a mineral solution suitable for plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms were discovered. As by omitting a particular element from the solution, a researcher can determine whether it is essential to the plant. So if the element left out of the solution is essential, then the lack of this element will make the plant appear abnormal when compared with control plants grown in a medium with complete nutrients. Most nutrient research involved crop plants and house plants, and little is known about the nutritional need of most of the uncultivated plants in the wild, as most of the application of hydroponic is in the near future is in farming business. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomatoes, seedless cucumbers, lettuce, green beans, cauliflower, cabbage, broccoli, lettuce, peas, spinach, carrot, beetroot, cucumber, onion, pepper, radish, strawberry, raspberries, blueberries, grapes, basil, coriander, and mint, etc. So let's now learn about the essential mineral elements. Most of the mineral present in soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some others gold, while some plants growing near nuclear test sites take up radioactive strontium. There are techniques that are able to detect the minerals even at a very low concentration. The question is whether all the diverse mineral elements present in a plant, for example gold and selenium, as mentioned earlier, are really necessary for plants. So now we would try to find out the criteria for essentiality. The criteria for essentiality of an element would be as follows. Number one, the element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete their life cycles or set the seeds. Second, the requirement of the element must be specific and not be replaceable by any other element. In other words, deficiency of any one of the elements cannot be met by supplying some other element. Third would be that the element must be directly involved in the metabolism of the plant. Now, based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. Number one, macronutrients, and number two, micronutrients. Macronutrients are generally present in plant tissues in large amounts. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from carbon dioxide and water, while the others are absorbed from the soil as mineral nutrition. So the above mentioned 9 essential elements, which are considered to be macronutrients because plants require them in relatively large amounts, 6 of the 9 macronutrients, that is carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus, makes up about 98% of plants' dry weight. The other 3 macronutrients, that is calcium, phosphorus, magnesium, makes up about 1.7% of plants' dry weight. Micronutrients. Micronutrients, or trace elements, are needed in very small amounts. These include iron, magnesium, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to the 17 essential elements, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Several micronutrients act as cofactors that cooperate with enzymes in catalyzing reactions. Micronutrients can generally be used over and over, so plants need only minute quantities of these elements. The requirement of molybdenum, for example, is so modest that there is only 1 atom of this rare element for every 60 million atoms of hydrogen in a dried plant material. Yet a deficiency of molybdenum or any other micronutrient can weaken or kill a plant. Essential elements can also be grouped into 4 broad categories on the basis of their diverse functions. These categories are: number 1, essential elements as components of biomolecules and hence structural elements of cells. Example, carbon, hydrogen, oxygen and nitrogen. Second, essential elements that are components of energy-related chemical compounds in plants. Example, magnesium in chlorophyll and phosphorus in ATP molecule. Third, essential elements that activate or inhibit enzymes. For example, magnesium is an activator for both ribulose biphosphate, carboxylase, oxygenase and phosphoenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zinc is an activator of alcohol dehydrogenase and molybdenum of nitrogenase during nitrogen metabolism. Could you name few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Fourth, some essential elements can alter the osmotic potential of cell. Example, potassium. Potassium plays an important role in opening and closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a cell. Essential elements perform several functions. They participate in various metabolic processes in the plant cells, such as permeability of cell membrane, maintenance of osmotic concentration of cell sap, electron transport systems, buffering action, enzymatic activity, and act as a major constituent of macromolecules and coenzymes. So now let us learn about some of the various forms and functions of essential nutrient elements. We'll begin with nitrogen. Of all the essential nutrients, nitrogen is the one that is most often limiting for crop growth. Nitrogen is the nutrient which normally produces the greatest yield responses in crop plants, promoting rapid vegetative growth and giving the plant a healthy green color. This is essential nutrient element required by plants in the greatest amount. It is absorbed mainly as nitrates, though some are also taken up as nitrites or ammonia. Nitrogen is required by all parts of a plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of proteins, nucleic acids, vitamins and hormones. So now we move on to phosphorus. Phosphorus is absorbed by the plants from soil in the form of phosphate ions, either dihydrogen phosphate or hydrogen phosphate. So we'll now move on to phosphorus. Phosphorus is absorbed by the plants from soil in the form of phosphate ions, either dihydrogen phosphate or hydrogen phosphate. Phosphorus is a constituent of cell membranes, certain proteins, all nucleic acids and nucleotides, and is required for all phosphorylation reactions. Phosphorus is a component of various enzymes and proteins. It is a vital component of DNA, the genetic memory unit of all living things. It is also a component of RNA, the compound that reads the DNA genetic code to build proteins and other compounds, essential for plant structure, seed production and genetic transfer. Phosphorus is essential for the general health and vigor of the plant. Some specific growth factors that have been associated with adding phosphorus to the crop are stimulated root development, increased stalk and stem strength, improved flower and seed production, more uniform and earlier crop maturity, increased nitrogen fixing capacity of legumes, improvement in crop quality and increased resistance to plant diseases. Potassium. It is absorbed as potassium ions, K+. In plant, this is required in more abundant quantities in the meristematic tissues, buds, leaves and root tips. Potassium helps to maintain an anion-cation balance in cells and is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and in the maintenance of the turgidity of cells. Calcium. Plants absorb calcium from the soil in the form of calcium ions. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membranes. It activates certain enzymes and plays an important role in regulating metabolic activities. It is also crucial in activating certain enzymes and to send signals that coordinate certain cellular activities. It is calcium that is key to normal root system development. Moving on to magnesium. It is absorbed by plants in the form of divalent magnesium ions. It activates the enzymes of respiration, photosynthesis, and are involved in synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Magnesium is needed by all crops to help capture the sun's energy for growth and production through photosynthesis. Photosynthesis takes place in the chlorophyll, the green pigment in plants, and magnesium is the central atom of the chlorophyll molecule, with each molecule containing 6.7% of magnesium. Now we'll talk about sulfur. Plants obtain sulfur in the form of sulfate. It is an essential element in forming proteins, enzymes, and vitamins in plants. Sulfur is present in two amino acids, cysteine and methionine, and is the main constituent of several coenzymes, vitamins like thiamine and biotin, coenzyme A, and ferredoxin. Iron. Of the micronutrients, iron is needed in the greatest quantity, and its availability is dependent on the pH of the growing medium. Plants obtain iron in the form of ferric ions. Iron is a constituent of several enzymes and some pigments and assists in nitrate and sulfate reduction and energy production within the plant. Although iron is not used in the synthesis of chlorophyll, the green pigment in the leaves, it is essential for its formation. It is an important constituent of the protein involved in transfer of electrons like ferredoxin and cytochromes. It is reversibly oxidized from Fe2 plus to Fe3 plus. During electron transfer, it activates catalase enzyme and is essential for the formation of chlorophyll. Manganese. Although it is used in very small amounts by plants, manganese is of capital importance to healthy plant growth. Like all micronutrients among others, it plays a significant part in the process of photosynthesis. Manganese is an important plant micronutrient and is required by plants in the second grade quantity compared to iron. It is absorbed in the form of manganese ions. It activates many enzymes and is involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Moving on to zinc. Zinc is one of the essential micronutrients and an important constituent of several enzymes and proteins. Is only needed by plants in small quantities. However, it is crucial to plants' development as it plays a significant part in a wide range of processes. Plants obtain zinc as Zn2 plus ions. It activates various enzymes, especially carboxylases. It is also needed in the synthesis of oxygen. Copper. Copper is absorbed as cupric ions. It is essential for overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reactions and is reversibly oxidized. Boron. Boron is not required by plants in high amounts, but can cause serious growth problems if it is not supplied at appropriate levels. It is absorbed as orthoborate like BO3 negative or B4072 negative, which is a tetraborate ion. Boron is required for uptake and utilization of calcium ions, membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Molybdenum is one of the essential micronutrients like manganese. Among other, it is only required in small amounts for normal plant development. However, it plays a crucial part in regulation of various plant functions. Plants obtain it in the form of molybdenate ions, molybdenum dioxide. It is a part of several enzymes including nitrogenase and nitrate reductase, both of which take part in nitrogen metabolism. It also needed by symbiotic nitrogen fixing bacteria in legumes to fix atmospheric nitrogen. Plants also obtain molybdenum to convert inorganic phosphorus into organic forms in the plant. Chlorine, it is absorbed in the form of chloride anion along with sodium and potassium. It helps in determining the solute concentration and the anion-cation balance in the cells. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. So to summarize what we have learned in this module, plants obtain their inorganic nutrients from air, water and soil. Plants absorb a wide variety of mineral elements. Not all the mineral elements that they absorb are required by plants. Out of the more than 105 elements discovered so far, less than 21 are essential and beneficial for normal plant growth and development. The elements required in large quantities are called macronutrients, while those required in lesser quantities or in trace amounts are termed as micronutrients. These elements are either essential constituents of proteins, carbohydrates, fats, nucleic acid, and take part in various metabolic processes. So learners, in the next module of this chapter, we'll be learning about deficiency symptoms of essential elements in plants. Till then, happy learning.